Hi, this is Karen from the University of Connecticut.
It's Brittany from Florida Southern College.
And Eric from State College of Florida.
So in this demo, we're going to try and walk through the many stages of the chemical extraction, which comes towards the end of the Small World Initiative semester. The chemical extraction procedure, generally speaking, is going to require 4 lab times. So it can take up to 4 weeks if you're meeting once a week, or 2 full weeks if you meet twice a week. But it's something to think about just in terms of spacing out and time management. So we're going to do our best to walk you through the different steps and stages so that you can kind of know what to anticipate with your results. So just to remind you, what we've done so far, hopefully, is that your students have done a spread patch and have isolated candidates that produce these nice clear zones of inhibition. And from those candidates, those environmental candidates, they've hopefully made it isolated colonies using a T-streak or quadrant streak or whatever you want to call it. So you'll hopefully have something like this where you have nice, clear, isolated colonies. There'll be some other activities you're doing in parallel. You might be doing your PCR analysis, et cetera, et cetera. But from these isolated colonies, what you're going to also do is make a nice thick lawn on LB media or whatever media it was that you have been growing your environmental isolate on. So I think in this situation, the student grew a lawn on LB media. And what we're going to actually do is use this lawn and the bacteria on it and use this in our chemical extraction. We're going to model the ethyl acetate chemical extraction procedure that is written up in the manual. There are other options outside of this, but this method has been fairly well tested and fits within that time frame. If you use other solvents, it might take even longer than the 4 lab periods. So we're going to walk you through these steps. There are always some modifications, but hopefully this will clarify what to expect. So what you're going to need for this essentially is your lawn of bacteria, a glass bottle. Okay, so everything that we're doing working with solvents is going to have to be in glass from this point forward. It's really important because if you use a plastic, your solvent is actually going to extract plastic. And so in that vein, if you use a glass bottle, be sure that you're never going to invert it into the cap unless you're working in some type of chemistry lab where you have the right equipment. Once you have, we're going to chop up our lawn of bacteria and agar and put it in the bottle. Once you have it in the bottle, you really want to make sure that it stays upright. This is also going to prevent any solvent from leaking out the sides because these aren't super tight seals. So because this is a little bit of can be a little bit of a messy procedure, what you want to be sure to do is be really careful about your clean space. And so when you are going to be doing this, you can work directly on the bench, but be sure to understand that you may actually spill some of this material on the bench, and you're going to need to take appropriate procedures to be sure to clean that up because this is an environmental sample and it is an unknown at this point. So what a lot of people do is they'll either use a paper towel in their workspace like this, or you can use bench coat or lab mat or whatever you might have available to you. And that way, if you do have any spillage, you can wrap that up and put it into your biohaz waste. Then, of course, you'd still want to spray down and wipe everything down in the environment that you're working with. Most people use metal spatulas for cutting up the agar in your Petri dish. And it's easier if you have more than one tool. It's tempting for the students to grab pieces perhaps with their hands, but there's lots of different approaches that you can take. We've had a student, we called it the appetizer approach, and she just used a toothpick and poked the pieces of agar and put it in the bottle. But essentially, whatever makes it easiest to get what we're going to make these small pieces of agar and get them into our glass bottle. So I'm going to go ahead and demo this now. So you're going to just basically cut your agar into small cubes. The larger the amount of surface area that you're providing to your chemical extract, the more likely you're going to get a higher concentration of chemicals out. So I'm just breaking through the agar. I did just like a grid, and then I'll turn it this way.
Karen, is there any need to somehow, not sterilize, but sanitize the spatulas before you actually use them, or is that unnecessary if it is?
So my assumption is that these were sterile, had come out of an autoclave, were wrapped in foil before we unwrapped them. So you do want to make sure that you're working with something clean, because hopefully whatever you're working with here is what you're extracting is just from your microbe. But all of this will basically be sprayed down and then cleaned afterwards as well. So if you're short on these, you can actually just clean them with a bleach in between and share in your lab. That should be enough, because I don't think there'll be a carryover effect in terms of the chemicals that you're getting out. The vast biomass is all of this on the plate. So what you'll see is that it can be a slow process, and now you'll just watch as if I have only one, you kind of have to go one cube at a time, and then it's sticky and it fights you to get it off into the bottom of the jar. I liked using these types of spatulas because you could kind of line them up like this in the groove of the spatula and then use both tools to just complement each other. So whatever works for you in your particular lab, you're just basically going to take all of these different cubes and get them into the bottom of your jar. Trying to not spill any in the surrounding lab space. Okay, so we've finished chopping up our agar, and you can see there's a nice empty petri dish. Nothing's left in the agar. The agar is sitting in this glass bottle. Just a couple of pointers. You want to make sure that when you're doing this, you want to use fairly small volume glass bottles, because the larger the bottle, the less effective it's going to be in terms of the ethyl acetate really getting in contact with all of the agar that you're putting in there. So the next step of our chemical extraction is that we actually need to freeze what's in the bottle. And the reason that we're freezing what's in the bottle is to actually break down the agar a little bit, to lyse the cells, to make all of the material there that we were trying to extract more accessible to the chemical solvent. So however you can freeze it is going to be what is up to you and your schedule. If you have a -80, you can put the bottle in the -80 for 20 to 30 minutes, and that's going to work great. If you have a -20 and you have longer time, and the suggestion in the lab manual is a dry ice ethanol bath, and so if you did have something like that made up, you could make it up in a container like this. This is empty for a demo, but you could make it up in a container like this, and then you would essentially have your students put the bottles in the container, and they kind of slosh it around in the container gently to make sure that the agar in the middle is also being frozen, and then maybe let them sit for 15-20 minutes so they're frozen all the way through. And you're going to take this step before you add your chemical extract.
All right, we'll see you in the hood.
Okay, so we're now continuing our chemical extraction. We've cut up our agar, we put it in a glass jar, and we've frozen it down either in a dry ice ethanol bath or at the -80. And so our frozen agar is now here, and the next step is to add ethyl acetate. So this step, a couple of things. This is definitely something to do in a fume hood, so you're going to want to be sure that you're working in a fume hood for the addition of ethyl acetate, and that everything you're working with is glass, including the pipettes that you're going to use. Our ethyl acetate has been aliquoted into a smaller working stock jar that you can see here, and I'm just filling up this glass graduated cylinder until I have 12 mils of ethyl acetate. So you should know that this is actually a rate limiting step in your lab, because you really don't want to have more than 1 to 2 students at a time, and most likely you or a TA should be distributing the ethyl acetate to the student and having them pour it into their agar. It definitely shouldn't be done unsupervised or outside of a chemical hood. So I'm just going to finish filling up my graduated cylinder here until we have about 12 mils. Okay. And there's a glass waste in the hood that you probably can't see. And we're going to add the ethyl acetate to our jar here. We're just going to pour that directly in. And the next step that we're going to do is we're going to actually add water to this chemical extraction. Those of you who are out there who are chemists, you may or may not feel that this is necessary, but we think that it helps the efficacy of the chemical extraction at this point. And then obviously you have 2 nice clear layers a couple steps down the road when we pull our extract off our agar. So I'm just going to use, this is sterile water that we have here, and I'm just going to use a pipette to pull it up, and we're going to add 8 mils of sterilized water. You add the water right to the bottle. Okay, and now we have started to extract our samples. What you're going to do now with your bottle is put it on an orbital shaker if you have one available to you, and you're going to gently shake your sample You don't, at this point, want to be really sure that you're not doing any inversion. Again, you have solvents in this bottle. And you can extract your agar for anywhere from 2 to 5 days. Less than that, you may not get a real effective extraction, although it's really going to vary. But when you start pushing it towards 5 to 7 days, if there is any activity, you might lose it. So most people actually will let extract from a Tuesday to a Thursday or a Monday to a Wednesday, or from Thursday overnight until when they meet again the following week. So you can keep it shaking at room temperature that entire time until you meet with your students again. And at that point, we will pull off our extract and look at the 2 different layers.
So if you also have an extended period of time where you may not be able to get back to your chem extract, you could also store this in the -20 or in the refrigerator, wrapped in aluminum foil to prevent any form of oxidation.
Great. Yep, that's absolutely true. So if you need to figure out a buffer in terms of your scheduling, you can just kind of pause everything by freezing it. Thanks. Okay. Okay, so we're now entering part 3 of our chemical extraction, which could have taken you different periods of time depending upon how you structured your class. And we're just going to show you a couple of examples of things that you might encounter as a result of your both the different types of media that you're going to use, as well as the different bacteria that might be growing on that media when you do the chemical extraction. So at this point, we have added our ethyl acetate in the last section, and then it was shaking for roughly 3 to 4 days, and your students can work in the hood to remove the extract from the bottle or at the bench, but at the bench, obviously be sure that in this entire section, the students should be wearing all of the appropriate personal protective equipment, including eyewear and gloves when you're working with the ethyl acetate along with the lab coat. So what you can see here are 2 bottles, and these bottles have been emptied into small 20 ml glass scintillation vials. These are definitely the easiest thing to use with the chemical extraction. So if you have access to the 20 ml scintillation vials, we highly recommend using these for this portion of the chemical extraction. But you can also use glass test tubes. Some instructors have used glass petri dishes. They present their own inherent challenges, but essentially any vessel you have that's glass will work if you can make it work. So all that the students have to do at this point now is using, again, a glass pipette, and you can, whatever your preference, long or short, the long do have a tendency to break more readily. They've actually gone in with their clean pipette and drawn out all of the ethyl acetate layer. So there's nothing left in here because it's all been drawn out and it's already been emptied into this scintillation vial. What we wanted to illustrate for you is that once you take the extract off of your agar and empty it into the scintillation vials, your extracts will have really different personalities, okay? And those are going to depend upon both the type of media that was used, that the bacteria were grown on, and also what bacteria were present. So what you can see here are 2 very different extracts. This first one has a very clear, very distinguishable boundary between our organic layer and our aqueous layer. Okay, so the ethyl acetate layer is on the top, and what you're seeing is a nice clear boundary. And so what the next step in this process is essentially going to be drawing that boundary that top layer off, because that is the only layer we're going to work with when we're testing going on to the next step. In this extraction, you can see that the layers did not separate nearly as clearly. There's a very small layer on the top, but really there's been a lot of mixing between those 2 layers. And in fact, there might be times that you'll see 3 layers or no layers at all. So some suggestions that you can do, obviously this is the best case scenario, but if you end up with a sample where you're not seeing a clear separation of layers, perhaps the easiest thing to do is to put that in the freezer. When you put that in the freezer, your aqueous layer is going to freeze and the organic layer will not, and then you can easily draw that off, and that's what you'll move forward with. So that's a really easy way to troubleshoot not getting really clear, distinct layers for your chemical extraction. So at this point, you're going to only be taking the top layer and putting that into a clean 20 ml scintillation vial. Okay? So again, using a clean glass pipette, you're going to draw off the layer, and you don't need to take the entire top layer. You want to try and not get that boundary between the two. And so the volumes here can also be really variable depending upon the amount of water that was in your plates when you started, the humidity in the air. So there will be some variation in terms of the volumes that your students are getting as they pull off this top layer, as well as the whole extract itself. Okay, so I'm going to leave, just like when you do, if you've done DNA extractions or anything like that, you want to leave, you're going to leave a little bit of that top layer behind. And now in this new clean scintillation vial, we just have what we have been seeking, which is our nice clean organic layer. And so it's going to be this layer that the students are going to be working with from this point forward. And the very next step that would happen at this point is you would actually make sure your vials are labeled on the side as well as the lid, and these vials would get placed in the hood and dried down. One of the great things about ethyl acetate is that within 2 hours, but definitely overnight, your sample will be completely dried down in this scintillation vial, and you'll be ready to move on to step 4. Is it up? So this is just one other addition to our chemical extraction, and a part of the course that students find really, really fun is that once you've taken that nice clean organic layer off the top of your extract, if you have access to a UV light or black light, it's really fun to shine this light on your samples and see all the rainbow of colors that you're going to see. So a lot of the molecules become activated. You could have greens and yellows and purples and blues. And it's a really great visual way to show the diversity, even though your extract is this clear liquid, that you might have some really different compounds in that extract. Okay, hi, we're just coming into the last phase of our chemical extraction, and we wanted to talk about a couple of questions that come up really often, which is how do we get rid of all this stuff that we've generated? So just to kind of walk you guys through, you've had your bacteria growing on a lawn and agar on a plate. So this plate obviously is going to go into your biohaz waste in your lab. Your glass pipette can just go into glass waste, obviously, but these are the things that are a little bit more questionable, which are the agar remnants in your glass bottles that still have solvent in them, as well as the aqueous layers of your extracts that you're not using. So the easiest thing to do with all of these is to take all of the caps off, let them sit in the hood, and let that ethyl acetate evaporate off. Once that is done, you want to take anything that you want to reuse. So such as these glass bottles, you would want to autoclave, and then that agar will get disposed in biohaz, and then you would clean and autoclave and reuse the glasses as needed. Some places might feel the need to reuse your scintillation vials. So here you would have to go through probably a pretty stringent wash cycle to just make sure that you're safely reusing them. But most people actually dispose of the scent vials because they are actually disposable and fairly affordable for that purpose. So hopefully that helps answer any questions in terms of what to do with your waste at the end of the chemical extraction. Okay, so we are now entering the last phase of our chemical extraction that we've done here at the Small World Initiative. And what we're going to actually do at this point, our organic layer has dried down in the hood at least for 24 hours. And what I just wanted to kind of illustrate here is that in my isolate, my extract here, you can't even really tell there's anything in the bottle at all. There's no apparent color. There's like a little bit of texture maybe. But this student's extract, you can hopefully see there's some real clear orange pigment that has been concentrated down as a result of that ethyl acetate drying down over that 24-hour period. So the next step is to actually resuspend this chemical extract. We're going to resuspend the chemical extract into just ethyl acetate again, and then we're going to use that resuspension, hopefully nice and concentrated, and we're going to dot that onto a plate, and we're going to test that against our tester strains, which we'll show you in another video. So for right now, we're just going to show you that how you're going to resuspend this. And at this point, it's probably easiest to aliquot your ethyl acetate into small bottles that your students can actually work with at their bench. You just want to be sure that, again, you're impressing that they're not doing any deep inhalations of the ethyl acetate, that they're using pretty good safety precautions in doing this. So what you're going to do at this point is essentially take 100 microliters of ethyl acetate out of your small jar, and we're just going to use a pipette to resuspend our chemical extract in the scintillation vial. So keeping in mind that our ethyl acetate is going to evaporate pretty quickly, you don't want the students to use a ton of time here. So I'm just gently pulling up the ethyl acetate and resuspending it by moving it around the scintillation vial. Once I feel that that's fairly well resuspended, I would prepare my plate that I'm going to use to actually test our tester strain against, and we will be sure to show you that stage in our next video. Hi, this is Karen from the University of Connecticut.
Brittany from Florida Southern College.
Eric from State College of Florida.
And we are here to finish up the last stage of our chemical extraction procedure. And in this stage of the chemical extraction procedure, we've gone through cutting up the agar, extracting with ethyl acetate, and then letting it dry down. And we had a dried extract that we showed you in the last video. And all we've done now is resuspended this extract with 100 microliters of ethyl acetate. So it's already been added to this container. And you can see it's a really small volume. But you can also see that it's a bright yellow color. So there's obviously something in there that's been nicely resuspended. This is now our concentrated chemical extract that we are going to use to test against an agar overlay of a tester strain of choice. So in order to set up for this experiment, what you're going to need to have is a small aliquot of ethyl acetate, which you can see here. This is just plain ethyl acetate. Your resuspended extract, but you have to work fairly quickly because this actually is going to evaporate off pretty quickly. And then we have an LB plate that we have labeled, you can see here, and the most important part of the labeling is that you have marked on your plate where you're going to dot your extract and where you're going to dot your control. In this situation, the control is our plain ethyl acetate. So all you're going to do at this point is use 10 microliters at a time. You're going to dot your extract onto your extract circle, your control onto your control circle, and let that evaporate off. You'll repeat that 3 times so that you have a total of 30 microliters of extract versus 30 microliters of your control dried on your plate. And then the next stage that I'm going to show you is making an agar overlay with a tester of choice. And as you can see, I have B subtilis on this plate, so we're going to use B subtilis as our tester. That's then going to incubate, and then we will be able to look at the inhibition of growth of the tester strain to see if our chemical extract shows activity. So I'll just walk you through that process. So I have a pipette set to 10 microliters. I'm going to take 10 microliters of my ethyl acetate. I'm going to dot this into my control. You want to be sure to not leave the ethyl acetate open for very long if you're not using it. And we're just going to let that evaporate off. As soon as that dries off, which should take a couple of minutes, we would add the second and then the third spotting of our ethyl acetate. For demonstration purpose, I'm just going to turn the plate and we're going to add 10 microliters of our extract to the other side. And you want to add this somewhat slowly so that it doesn't spread too far onto your plate. And that air bubble will pop off as it dries. Okay, now we would just wait for that to dry, and we would repeat that 2 more times again, so you have 30 microliters of extract, 30 microliters of your control, and then you'll be ready for your next step, which is to pour the agar overlay. Okay, so we're going to finish up our final stage, we think, of the chemical extraction. So as I showed in the last video, we've dotted our plate so that you can now see that there's an orange hue to where our extract is, and it's dried. And then you normally won't really see too much in the control region because that's hopefully just been your ethyl acetate that has evaporated off. You want to be sure with these plates that you actually space these pretty far apart. You don't want to put them too close because that can actually complicate the interpretation of the results. So at this point we're getting ready to make our agar overlay. The way that we're going to do it is we're going to use B. subtilis as an overnight culture. I'm going to take 100 microliters from our B. subtilis and add it to a 15 mil centrifuge tube. And then I'm going to add 7 mils, and this is hot augur. So this is a 50% LB augur, and we just microwaved it. So you need to make sure this isn't too hot, that it's going to kill your bacteria when you mix the two together. Many schools might have your soft augur actually in a water bath in your lab, and that's a really easy way to just have them already aliquoted out into 7 mil tubes. And if that's the case, you would take that, add the bacteria after the fact, and then vortex it. I think the key is to make sure that you have a good even distribution of the bacteria in your augur. So here I'm just going to add 100 microliters to the bottom. And 7 mils Okay, I'm going to give that a vortex. And then what you want to do is pour this quickly onto the plate, not on either area where the extract is, and then just quickly, I'll try and not get my hands in the way, quickly tilt it so that it covers the whole surface area of your plate, just like that. Then you don't want to disturb the plate. You want to let this solidify in place. So for about 5 minutes for sure, make sure that the plate is not disturbed. Once that augur has solidified, you can then incubate at whatever temperature you've been maintaining your cultures at. Okay, so here we are. We're ready to look at our actual data, the actual results that came out of our chemical extraction that you have spent the last 4 sessions working through with your students in theory. And we have some really great examples of positives and also of some that didn't really work. And it's kind of interesting when you look at all the pieces together. So this is just reminding you of a streak plate, and this is showing you that this isolate here had activity against B. subtilis. So this is the isolate that was then brought forward, and we did our chemical extract, and you can see really clearly that against the lawn of B. subtilis, there's a beautiful clear zone of inhibition where our chemical extract was added, and there's obviously not any negative effect of our control. And so this is an example where you're seeing that the activity is mirrored in this assay that you're seeing when you have the activity against in your spread plate. But interestingly, that same isolate did not show any activity of the chemical extract against E. coli. So again, you're going to have varying results depending upon the tester strain that you're using in your augur overlay, as well as the initial bacteria that you're working with. Just to show you some other really nice examples of the results of a chemical extract. This is from another isolate with another student, a nice clearing here, and the control showing not as much of a clearing. This same sample against a different tester strain, a smaller clearing or zone of inhibition, and again, no effect in the control group. So you can see that you can have a range of results from no effects at all to some really visually stunning zones. And so this is a great opportunity to talk to your students about why you're seeing what you're doing, and then bringing forward their thinking processes.